Okay, in this lecture, we are going to focus on something that is easy to overlook. That is where data actually comes from. Before you ever train a model, biological data passes through a long pipeline from a scientific question to an experiment and then to a raw signal and then to files that eventually land on your laptop. If you don't understand that story, it is very easy to build models that look good on paper but on computer but fail in the real world. Let's start discussions on this story from a question to file. So we are looking at how biological data actually become a file you can model. A scientific question drives an experiment. You know this, right? And then that experiment produces a raw physical signal. And pre-processing turns that signal into the file an ML team eventually sees. So this is also where scientists and data engineers overlap. One designs the assay and generates the signal. The other manages, transforms, and stores the data. Different biological scales produce different kinds of files. At the molecular level, you might see smile strings or sequencing reads. At the cellular level, images are omics tables, and at the clinical level, structured tables alongside free text notes. Um, Finally, it is important to recognize that um, pre-processing decisions and missing or incomplete metadata introduce real risks like batch effects or feature leakage. If something goes wrong upstream, the model may learn the wrong thing. Okay, so watch out. Every dataset starts as a question. So that question drives an experiment, and the experiment produces a raw physical signal. And only after pre-processing does that the signal becomes the file you eventually see. If you look only at the file and not the steps that came before it, you miss the assumptions, constraints, and biases that are already baked into the data. The goal here is to trace that um, upstream story. So when you build a model, you understand not just what the data look like, but why they look that way. Now let's start, these two roles, scientist perspective and data engineer perspective. Um, let's start with a scientist's role in this pipeline. The scientist begins by designing a specific experiment to test a single hypothesis, let's say. They were close to the instruments and see the raw, unfiltered data as it is generated, often before any heavy pre-processing. Their analysis usually ends with something interpretable like a figure or a summary table rather than a reusable dataset. And most importantly, they interpret trends in biological context, asking whether the result makes sense, with that underlying biology. Now let's see an engineered emphasis. On the other side of the pipeline is the data engineer. Their goal is to build models that generalize across many experiments, not to analyze any a single one in isolation. So they typically work with exported processed files, maybe CSVs or TSVs rather than raw instrument files. By the time the data reaches them, key filtering, normalizations, summarization decisions, all those things are already made. Their job is to quantify and model those patterns computationally, often without a full visibility into the upstream experimental choices. So let's see. I mean, this is important, right? So both needs to speak with each other. Now let's see a typical data hierarchy. It helps to think about biological data as a hierarchy. At the bottom is raw data. This is the direct output of an instrument before any cleanup or interpretation. Next is pre-processed data. This is where signals are cleaned, normalized, aligned, or summarized. So they are useful for analysis. At the top is interpreted data. This is where domain knowledge is applied, turning processed signals into biological or clinical conclusions. Most machines, most machine learning models only ever see the top layer, but the limits of what a model can learn are set much earlier during pre-processing. Once information is discarded, it is generally gone. So be careful. Let's see some examples of this data hierarchy. Now let's see some examples. In genomics, raw data might be FASTQ reads straight from the sequencer. Processing aligns those reads into BAM files. Interpretation turns them into variant calls, for example. In imaging, raw data is a TIFF image from a microscope. Processing extracts cell features. Interpretation assigns phenotype labels. In clinical data, raw inputs are maybe lab values over time. Processing standardizes them into tables. Interpretation produces something like a risk score. Each step simplifies the data, but each step also removes information. Understanding where your dataset sits in this hierarchy tells you what questions your model can and cannot answer. Let's see a sequencing data example. At the molecular scale, the question is, maybe a simple question. Does this patient's genome contain a mutation linked to a disease? To answer that, biologists collect cells, isolate DNA, and prepare sequencing libraries, which are then run on a sequencer. The sequencer detects fluorescent or electrical signals that are converted to base calls through computational base calling. Every modeling file you will see depends on how those signals were generated and processed. So, missing this context can lead to the wrong conclusions. Models rarely see raw sequencing reads, for example, but they inherit every assumption made before those reads were turned into features. Okay, let's see these evolving files concept. So sequencers, they do not read letters. I mean, they read signals, right? And then base calling converts that signal into FASTQ file plus per base quality scores. Then we align reads to a reference genome to uh, to get a BAM file, and that step can introduce reference bias. For example, structural variants or population specific alleles may be missed if they are absent from the reference genome. Finally, a variant calling produces a VCF file, but it is driven by thresholds, like depth and quality. If you model only the VCF without understanding, you may miss uncertainty in downstream processed files. Depth information may be discarded, making high confidence and low confidence variants, indistinguishable. Now let's see the cellular scale. So for the a cellular scale, maybe an example question could be, does a treatment change how cells behave? So biologists culture cells, apply treatments, sustain different cellular structures. Example, nucleus, so DAPI, cytoplasm, organelles, and image the plates using automated high content microscopes. That process generates tens of thousands of images. Those images are then segmented and quantified, turning pixels into per cell measurements like size, shape, intensity, and texture, for example. By the time this reaches you as a CSV file, the raw images are gone. You are looking at maybe, I don't know, millions of rows of engineered single cell features. That spreadsheet is not the experiment. It is the output of a long image processing pipeline with a lot of assumptions at every step. So let's see that data transformation. So let's see imaging transformation from signal to TIFF. At the signal level, the microscope is just detecting fluorescence. photons emitted by different dyes, typically one channel per marker. Those signals are stored as TIFF images, capturing pixel intensities across the field of view. Even at this stage, experimental choices matter, and then also exposure time, illumination drift, focus. So all these differences, and then across plates can produce batch effects before any analysis even begin, right? And then from a TIFF to CSV. So next, image analysis software segments individual cells and computes features like area, shape, intensity, texture, and more. Every feature depends on segmentation quality. If cells merge, split incorrectly, or debris is misidentified as a cell, those errors propagate directly into the feature table. By the time the data becomes a CSV, those mistakes are, pretty much silent. There is no flag that tells you your cell was poorly segmented. So finally, scientists or software assign phenotypic labels such as normal, apoptotic, or mitotic, those kinds of labels now. So though these labels uh, become your ground truth, but they are only as reliable as the upstream segmentation, feature extraction, and sampling decisions, which are usually invisible once you are working with the CSV file. Now let's take electronic health records example. At the clinical scale, the question shifts from how a system behaves to which patients benefit and why. Unlike the lab, there is no controlled experiment here. Data is generated during routine care across hospitals and clinics. And then again, not under standardized conditions. Clinicians document lab results, and there is some diagnosis, and there are medications and procedures and outcomes. All those things are in electronic health records. Over time, each patient becomes a longitudinal data stream combining structured fields with unstructured clinical notes. This data is highly heterogeneous. Different hospitals use different software, coding systems change, units are not always consistent, and free text notes are noisy and incomplete. And there is a strong selection bias. We only observe patients who interact with the healthcare system, often after something has already gone wrong. So let's see how EHR data are formed. So here, from the signal to raw EHR. EHR data begins as a mix of structured measurements like I don't know, glucose levels or white blood cell counts, and unstructured clinical notes, clinician notes. These data are generated across many clinical systems, prescriptions, procedures, lab results, and imaging. All these things often live in separate databases and must be linked using patient identifiers, timestamps, and encounter IDs. Even at this stage, the data is fragmented and inconsistent across hospitals. So from raw to processed, right? And then cleaning EHR data is usually the most time-consuming step. Units can differ, coding systems evolve, timestamps may be incorrect, and missing data is, there always. Critically, missing data can be ambiguous. If a lab value is absent, you cannot tell whether the test was never ordered or was not necessary or simply wasn't recorded. That uncertainty gets baked into the processed tables, right? So from processed to cohort, to build a modeling dataset, you define inclusion and exclusion criteria and extract a cohort. This step introduces a substantial selection bias. For example, requiring complete lab panels preferentially selects sicker patients who interacted much more with the healthcare system. The resulting cohort reflects clinical decision-making patterns, not exactly the underlying population you might want to generalize to. All right, now let's see, why story matters. This table is in our summary for entire lecture. At every biological scale, data looks clean by the time it reaches a model, but each step hides irreversible transformations. At the molecular level, base calling, alignment, variant filtering, all these things can introduce reference bias and false confidence. At the cellular scale, segmentation and thresholding shape the features you treat as ground truth. At the clinical level, normalization and text parsing encode clinical behavior, not just patient biology. None of these risks are visible in the final dataset. They only make sense if you understand how the data was generated, processed, and interpreted. That's why robust modeling in biology starts with the data story. Let's see some questions you need to ask. When someone hands you a CSV, these are the first questions to ask. What experiment did it come from? What filtering has already happened? How many samples were lost at each step? Are there batch effects? What do the labels actually mean? And what's missing? And why? If you cannot get clear answers, you are likely to learn pre-processing artifacts or batch effects instead of biology. That's why talking to the scientists is not optional. You need enough context to decide whether the data set is even, a modelable. Thank you.